DNA mutation. So DNA mutation means the permanent change in the nucleotide sequence of the DNA, and it can happen due to various reasons such as error in the DNA replication, chemical damage, or radiation. Whereas the DNA damage refers to the temporary changes or the disruptions that occur in the DNA structure. DNA mutation are the permanent one and DNA damage are the temporary changes that occur in the DNA structure. It can be caused by environmental factors like radiation and chemical and the normal processes inside the cells like the metabolism. Types of DNA damage and their mechanism. There are various types of DNA damage that can happen inside the DNA due to normal cellular process or exposure to the damaging agents which are present in the environment. First one is the DNA strand break. DNA strand break happen when one or both the strands of the DNA are cut. It is of two types, single strand break and double strand break. Single strand break is when one strand is cut and double strand break is when both the strands are cut. The causes are the ionizing radiations and the chemicals. Most of the causes are the same. The oxidative damage can occur due to the action of reactive oxygen species like hydroxyl radicals. These molecules react with the DNA and causes the base to damage, leading to the formation of lesions. Third one is the alkylation of the DNA bases. There are several alkylating agents, both endogenous and exogenous. Endogenous means from inside of the body and exogenous means outside of the body. So alkylating agents, both endogenous and exogenous, can modify DNA bases by inducing the alkyl groups. These changes can be cytotoxic, which means toxic to the cell, and cause mutation or have no effect. Now, base loss. Base loss happens when a nitrogenous base is simply removed from the DNA. This leaves behind a basic site, which is also known as AP site. A basic site simply means apurinic or apyrimidic site. AP sites are chemically unstable, which leads to mutation. Fifth one is the bulky adduct formation. It is caused by chemicals like polycyclic aromatic hydrocarbons (PAHs). These chemicals covalently bind to the DNA bases and form bulky adducts. These adducts disrupt the DNA structure, block replication, transcription, or other repair processes, and can cause mutations. Sixth one and the last one is the DNA cross-linking. It occurs when two DNA bases link together by a covalent bond. It is of two types, intrastrand or interstrand. Intra-strand crosslink means the link is within the same DNA strand, and intra-strand crosslink means the link between opposite DNA strands. And this crosslink prevents the separation of DNA, which is essential during the process of replication or transcription, leading to the disruption of the basic cellular processes. Sources and agents of the DNA damage. DNA damage can happen due to endogenous or exogenous factor. Endogenous means which happen inside the body, exogenous means which happen outside the body, which may damage the DNA structure and its function. So endogenous DNA damage includes replication error. It occurs during DNA replication when wrong nucleotides are added. So replication error is caused by DNA polymerase which has low accuracy. This mistake may remain in DNA if not corrected. Second one is the topoisomerase activity. So topoisomerase are the enzymes that removes the DNA supercoils during replication or transcription. However, the misalignment of the DNAs can stabilize the topoisomerase and DNA cleavage complex, which result in the formation of lesions. Third one is the reactive oxygen species. Reactive oxygen species are actually produced during the cellular processes and can cause oxidative damage to the DNA. स्मॉल अमाउंट्स नॉर्मल होते हैं लेकिन ROS अगर ज़्यादा अमाउंट में हो तो वो DNA को डैमेज कर सकता है. Excessive ROS amount can cause base modification, strand break or lesions. It is also associated with several human diseases like cancer, Alzheimer's and diabetes. And the last one is the endogenous alkylating agents. These agents can add methyl or ethyl group to DNA bases, leading to chemical modification. इसका example is spontaneous methylation of the DNA. Some modified bases can be mutagenic and lead to permanent changes in the DNA sequence. Exogenous DNA damage, ये वो डैमेज होता है जो कि एक्सटर्नल एजेंट से होता है, जैसे केमिकल्स, रेडिएशन, पॉल्यूटेंट्स. ओके, फर्स्ट वन इज द आयोनाइजिंग रेडिएशन, जिसका एग्जांपल है एक्सरेज एंड गामा रेज. ये डायरेक्टली डीएनए को ब्रेक कर देता है और जनरेट हाईली रिएक्टिव हाइड्रोक्साइल रेडिकल. अब रेडिकल से ऑक्सीडेटेड डीएनए डैमेज होगा. अब आयोनाइजिंग रेडिएशन कैन कॉज बेस डैमेज एंड सिंगल स्ट्रैंड और डबल स्ट्रैंड ब्रेक. Second is the UV radiation. UV radiation from sunlight is the major source of skin cancer. अब UV radiation का सबसे बड़ा इफेक्ट है वो pyrimidine डाइमर्स को फॉर्म करता है. pyrimidine डाइमर्स क्या होते हैं? टू pyrimidine bases on the same strand which are bound together, उनको pyrimidine डाइमर्स बोलते हैं. और ये डाइमर्स रेप्लिकेशन और ट्रांसक्रिप्शन जैसी प्रोसेसेस को ब्लॉक करते हैं. That's why UV radiation is very harmful for our body. Exogenous alkylating agents. Exogenous alkylating agents are found in tobacco smoke and industrial chemicals. So that's why हमें स्मोकिंग नहीं करना चाहिए, क्योंकि उसके जो exogenous alkylating agents हैं उसके अंदर जो agents present हैं वो हमारे डीएनए को डैमेज करते हैं. ये क्या करते हैं? अल्काइल ग्रुप को ऐड करते हैं इन द नाइट्रोजिनस बेस इन डीएनए व्हिच कॉजेस द कैंसर. इसका एग्जांपल है सल्फर मस्टर्ड्स एंड नाइट्रोजन मस्टर्ड्स. फोर्थ वन इज द एरोमेटिक अमाइड्स. अब ये भी सिगरेट स्मोक में होता है, डाइस में होते हैं, कोल में होते हैं एंड पेस्टिसाइड में होते हैं. ये लेशंस क्रिएट करते हैं इन द डीएनए व्हिच कैन लीड टू बेस सब्स्टिट्यूशन एंड फ्रेम शिफ्ट म्यूटेशन. Last one is the polycyclic aromatic hydrocarbons. This causes the formation of bulky adduct. ये वापस टोबैको स्मोक में होते हैं, व्हीकल एग्जॉस्ट में होते हैं एंड पॉल्यूटेंट्स में मिलते हैं. ये जो पॉलीसाइक्लिक एरोमेटिक हाइड्रोकार्बन है, लीवर के एंजाइम से एक्टिवेट होते हैं एंड आफ्टर द एक्टिवेशन दे फॉर्म रिएक्टिव सब्सटेंस दैट डैमेजेस द डीएनए.